Hi kids, so our discussion for today is all about carbohydrates, and this will be our first topic for your midterm period. So of course, when you get to hear the word carbohydrates, the first thing that you would think of are actually this kind of food. Diba? From your pizza, your pasta, to your burger, or your donut, or even your fries and your cake. Because generally, when we talk about carbohydrates, these are energy-giving foods. So what are carbohydrates exactly? From the word carbohydrates, this means that these are hydrates of your carbon, meaning to say that your carbon is being paired with your hydrogen and including your oxygen. Now, carbohydrates comes from the word saccharum, which means sugar, and it is derived from the formula CN, H2O, and then letter M. So meaning we are looking at how many carbons are present, how many hydrogen and oxygen are present. So one example would be your glucose having 6 carbons, 12 hydrogens, and 6 oxygen. So it could be written down in this form, or as stated here, it could be presented also this way. So C6H2O, there are 6 of your H2Os. Now, saccharides are simpler members of your carbohydrate family, and saccharide is considered to be the unit structure of your carbohydrates. So, how important are carbohydrates? Now, carbohydrates is your most abundant dietary source of energy, especially in the form of your glucose. It also serves as a storage form of energy in the form of your glycogen. So meaning to say you'll get to learn how your glucose are converted to glycogen and how glycogen is also being converted to glucose depending on what the body needs. Syempre po, if there is excess glucose, then we will not actually waste or just remove the excess glucose, but instead, para may reserve po tayo, then we will convert it to its storage form, which is your glycogen. And if in the case that all of the glucose that is present in our body is already exhausted, then that's the time that we will convert glycogen to glucose, and then there is still enough energy to be used all throughout the day. Now, carbohydrates are also precursors for many organic compounds, such as in the making of your fats and amino acids, and that it also participate in the structure of your cell membrane and your cellular functions. So recall our discussion about the cell. We pointed out the presence of your glycolipids and your glycoproteins, and those are examples of carbohydrates linked to another type of substance. Next, now certain carbohydrate derivatives are used as drugs like your cardiac glycosides or even your antibiotics. So this importance that we have discussed can actually be summarized in this way. So again, carbohydrates are intermediate energy. They are also intermediates of metabolism. They are for storage of energy. They constitute or part of your nucleotide in the making of our DNAs and RNAs. And lastly, they are found or they also serve as part of our structure or structural component of your cell. Now, how are carbohydrates named? In terms of the nomenclature of your carbohydrates, it will always end in OSE. So just like your enzyme na it ends with ASE, for carbohydrates that is OSE. And in naming, in the total naming uh of your carbohydrates, follow lang pa rin po natin just like what we discussed in your introduction to biochemistry na yung pag-count ng carbon atoms nyo. If tatlo, then that is tri. If apat, then that is tet. If lima, then that is pent. And then we have your hep, your hex, and so on and so forth. So, kunwari may tatlong carbon atoms tayo, in that carbohydrate, then we call that triose, then tetrose, then pentose, and so on and so forth. Now, in classifying your different types of carbohydrates, they are classified on the basis of carbon atoms they possess, or it could be in terms of the two functional group that is present in their structure, which could either be an aldehyde or a ketone group. So here in this image, this already summarizes the classification and nomenclature of your carbohydrates, of which we will get to discuss each one of these. Basta na-mention po natin yung first two kanina, ito po yun under number of carbon atoms and the type of functional group that is present. So we'll get to discuss further the rest. So, unahin natin yung classification of carbohydrate on the basis of molecular size. In terms of looking at the molecular size, we have your monosaccharides, your oligosaccharides, and your polysaccharides. Actually, almost the same din po ito, yung definition niya, dun sa discussion po natin sa amino acids and protein. Siyempre, when we say mono, isa, and then in terms of your oligo and polysaccharides, so may tatandaan na lang po tayo in terms of the number of units para masasabi natin na it is under your oligo or is it already under your polysaccharide. So let us begin with your monosaccharides. Now, what are monosaccharides? Now, monosaccharides are your simplest sugar. It is considered to be your simplest sugar because it cannot be hydrolyzed further into smaller units. Na kumbaga siya na talaga yung ending product natin. So, syempre, most of the food that we eat, yung mga carbohydrate-rich food that we usually intake are actually polysaccharides, and that is why there is digestion because we need to break down your polysaccharides because those are very big units, and ang ending niya is presence of monosaccharides. So an example of monosaccharides is your C6H12O6, and that your monosaccharides contain only a single carbon chain. Now, monosaccharides are further classified in terms of the number of carbon atoms, and monosaccharides have 3 to 9 carbon atoms. So we will base already the name of your monosaccharides on the number of carbon atoms that is present. So as stated a while ago, monosaccharides are classified on the number of carbon atoms, and the smallest monosaccharides would be having 3 carbon atoms, and that is what we call your trioses. So as you can see here in this image, ayan po yung tatlong carbon atoms po natin, and under your trioses is your glyceraldehyde. So glyceraldehyde, if you write down its number of carbon, hydrogen, and oxygen, so that would be C3H6O3. And in terms of the role of your glyceraldehyde, it is actually an intermediate in respiration, in photosynthesis, and in other branches of carbohydrate metabolism. The one that you see here inside a box emphasizes the importance of your glyceraldehyde in order for us to create your triglyceride, which is an example of a lipid. Lipids will be our next topic, and that without your glyceraldehyde, we will not be able to get glycerol, which is an important component of your triglyceride. So, kumbaga, glyceraldehyde, i-convert to glycerol, and then glycerol added along with other substances pa in order to form your triglyceride. So that is how important your glyceraldehyde is. Next, we have your pentoses having 5 carbon atoms. So as you can see here in this image, it is just presented differently. Ito yung cyclic form. Itong andito kids is yung straight chain nyo. So mas madali mag-count ng carbon atoms in a straight chain. But as to your cyclic chain, your first carbon will always start at this point. There are two examples of pentoses. You have your ribose and your deoxyribose. The main difference of ribose and deoxyribose, as stated here, is that your deoxyribose has one oxygen atom less than your ribose. And at what specific portion nakulangan ng oxygen si deoxyribose? So here, as highlighted, sa second nyo na carbon, wherein sa ribose, OH yung naka-attach. Sa deoxyribose, hydrogen lang. Now, as you all know, ribose and deoxyribose are important part of our RNA and DNA. Ribose for your RNA, deoxyribose for your DNA. So, your pentoses are important in the synthesis of your nucleic acid. So, recall the meaning of your RNA and DNA. Diba, RNA, ribonucleic acid, DNA, deoxyribonucleic acid. Now, other than for the synthesis of your nucleic acid, ribose is also needed in the synthesis of your coenzyme. So remember our discussion as to what coenzymes are. But these are cofactors. These are considered as your helper molecules in order to activate your apoenzyme. And these are examples of your coenzymes, your NAD, your NADP, and then included na din po CFAD. So looking at the structure of these different coenzymes, ayan po kasi si ribose nyo. Itong cyclic na nakikita nyo dito. So that is why ribose is important in order for us to create your coenzymes. And then your ribose is also important in the formation of your energy. Diba? Remember your ATP, that is our energy source, your adenosine triphosphate, in which it can also become your ADP or your AMP. And the only difference between the three is in the number of phosphate group that is attached to your ribose. So next we have your hexoses. Hex, so meaning there are 6 carbon atoms present here, and glucose is actually under your hexoses. Other than glucose, you have your fructose and your galactose. So looking at your structure here, so again, ayan po yung first carbon unit nyo. Medyo kakaiba lang po in terms of your fructose because of the type of functional group that is present in its structure. So glucose, fructose, galactose, this can all be sources of energy, especially in respiration. Glucose is the most common as a respiratory substrate and the most common monosaccharide. Siyempre, glucose po talaga yung ating pinaka-main source of energy and what is really being utilized by our cells. But in the case wherein there is no longer glucose that is present in our body, like totally nag-zero na kids, wala na rin po tayong energy reserves, wala na rin po tayong yung glycogen, yung ating energy reserves para ma-convert to glucose, then that's the time that we could make use of your galactose and fructose. They can actually also be used as energy. They can be converted para meron pa rin po tayong energy na magagamit in our body. But then again, that is already parang pinaka-last na resort na ng ating body. Now, as to your fructose, fructose is very common and it is found in the nectar of flowers. Fructose is actually one of the sweetest of all our natural carbohydrates. Now, hexoses are needed to synthesize both your disaccharides and your polysaccharides, especially your glucose. Glucose is particularly important because there are polysaccharides kids na tanging glucose units yung andun sa kanilang structure. Then again, ma-explain ko lang din po yan further once we get to that certain part. Basta again, hexoses are needed to synthesize both your disaccharides and your polysaccharides. Now, ito pong andito, these are already examples of your disaccharides. You have your sucrose, your maltose, and your lactose. Pero dili sa ako mag-una-una, Again, when we reach this portion here in our discussion, then you get to understand what this means. So, padayon sa ta. Now we go to the classification of your monosaccharides according to the functional group that is present. Now, the functional group that is present in your monosaccharides are actually carbonyl groups, meaning ito po yung mga functional group that is composed of a carbon atom that is double bonded to an oxygen atom. And there are only two. You have your aldehyde and you have your ketone or your keto group. Looking at the image, ayan yung aldehyde group nyo. So, ayan yung double bond O. Ito po yung ketone group nyo. Ayan po yung double bond O. So, those monosaccharides that have an aldehyde group would be called as aldoses. Those na may keto group are called ketoses. Now, it is very easy to identify if aldehyde or keto group ang functional group na present, if they are aldoses or ketoses, because your aldehyde group will always be at the endmost portion of your straight chain, whereas your keto group will always be found here at the middle part or at the second carbon. So, yan yun always, kids. Kumbaga, this is true to all na laging nasa end part si aldehyde and laging nasa middle portion si ketone group nyo. So, this is the examples of aldoses and ketoses, depending also of the number of carbon atoms that is present. So, we've made mention about glyceraldehyde. Glyceraldehyde is actually under your aldoses. And yung, if ever ketone group man, yung present sa ating triose, then we call it your dihydroxyacetone. There are some book, kids, na instead na de, ang nakasulat is di. So, they are just the same. Pareho lang po sila. So, we can either make use of dihydroxy or dihydroxyacetone. And then, looking at dito po sa hexoses nyo na part, si glucose, si galactose are actually examples of aldoses, while your fructose is actually a ketose. So, meaning ketone nag-functional group yung present kay fructose. So, that is actually the reason why when we draw it in its cyclic structure, different yung kanyang itsura because it has a different functional group compared to your glucose and your galactose. So, looking at the structure of each, so ayan, as mentioned, always yung aldoses nyo, yung double bond O nasa pinaka-end portion, nasa first na carbon, and always for your keto group, nasa second carbon or middle part, makikita yung double bond O. Now, there are three different structures of monosaccharides. There are three ways that your monosaccharides can be written down or its formula or structure can be drawn. So, dalawa po yung yung na-observe nyo kanina, diba, yung straight chain nyo, and that straight chain is actually what we call your Fisher projection. For your cyclic or ring structure, that is what we call your Haworth projection. And then we also have your boat and chair form, and that is what we call your X-ray diffraction analysis. Although here in our discussion, we will never be talking about X-ray diffraction analysis na form niya, yung pinaka-important po talaga and what is emphasized in your reference books is yung Fisher projection and yung Haworth projection nyo. So you'll get to understand better later once we discuss your different isomers kung bakit kailangan natin ma-identify na dapat straight chain siya, dapat we should look at its cyclic form, kasi that depends on the type of isomer that we are talking about. There are types of isomer kasi na dun lang natin ma-identify once we look at its ring structure or once we look at its straight chain. So again, this is your straight chain. Ayan, this is your ring structure. And then this is your X-ray diffraction. chair form daw siya. So, ayan siya, kids. So, now, let us discuss isomerism. So, since dun pa sa ating discussion, I think sa amino acids and protein, di ba? I think ilang beses na natin na-touch yung term na isomer. I think sa introduction pa nga, introduction to biochemistry, di ba may isomerism na tayo na na-discuss or yung tautomerism nyo. So, going back to what isomer meant, these are compounds possessing identical molecular formula but different structure. So, meaning they could have the same number of carbons, hydrogen, and oxygen, but once we draw their straight chain ba or cyclic na structure, may makikita po tayong differences sa position ba ng hydrogen or oxygen. And there are actually various types of isomerism. It could be structural or it could be your stereoisomerism. So start muna tayo with structural isomerism. So here, same ang molecular formula, but differ from each other by having different structure. So one best example here would be your glucose and fructose. Diba remember, they are both hexoses. They both have the same chemical formula na 6 kabuok ang carbon, 12 kabuok ang hydrogen, and 6 kabuok ang oxygen. But then, when we try to observe their structure written in its straight chain form, since fructose is a keto group, thus dito po sila nagdi-differ in terms of the position of their double bond. So, isomer sila, pareho po sila ng molecular formula, pareho ng number ng carbon, hydrogen, and oxygen, but as to the structure, magkaiba because they have different functional groups present. That is what we mean by structural isomerism. How about your stereoisomerism? Here, same ang molecular formula, same ang structure, asan sila nag-differ in terms of the configuration. So here, there is presence of asymmetric carbon atoms that allow the formation of your stereoisomerism. Asymmetric carbon, kids, is the same as saying chiral carbon. Diba naalala nyo yung ating definition about chiral carbon, wherein your carbon atom is attached to 4 different types of atoms or groups of atoms. Yan yung naka-unique din po kay amino acids, 'di ba? With the exception lang nung isang amino acid natin. So, again, because of the presence of an asymmetric carbon atoms, it allows the formation of your stereoisomerism. And there are actually 4 different types of stereoisomerism. You have your DNL isomerism, you have your optical isomerism, you have your epimerism, and you have your alpha and beta anomerism. So, start with DNL isomerism. Now, DNL isomerism, we call them enantiomers. These are chiral molecules that are mirror images of one another. So from the term mirror images, meaning to say, when you try to flip it back, diba? Parang when you try to flip it back, the same talaga yung structure, pero naka-mirror image lang siya, parang naka-flip back. And there are 2 types of enantiomers. It can be represented by your letter D and your letter L. So when you write this down, kids, capital letter po talaga dapat yung letter D and yung letter L nyo. Now, D monosaccharide has your OH group on its penultimate carbon found at the right. L monosaccharide, yung OH group on its penultimate carbon found at the left. So what do we mean now by penultimate carbon? Penultimate carbon is your second to the last carbon. Again, penultimate is your second to the last carbon. So, whenever we count your carbon, it will always start on top, and we can only identify your D and L isomerism sa straight chain. So, ayun. Kaya kung makikita nyo, may counting na po dito and always yung number 1 or yung first carbon nyo, yung pinakauna sa pinaka-top. So, saan yung penultimate? Second to the last. So, here, given that you only have 3 carbon, thus, yung second carbon nyo, of course, second to the last, then that is where we are going to identify if it is letter D or if it is letter L. Here, may 6 carbon po tayo. Again, second to the last. So, dun tayo mag-check sa fifth na carbon nyo. So, as highlighted here, ayan na po yung position ng OH nyo. Again, if your OH is on the right side, then that is letter D. If OH is on the left side, then that is letter L. So, very easy to remember. L would already immediately mean left. And then, of course, yung D nyo na agad is si right. So, kaya kung makikita nyo dito, may letter L, letter D, because again, nasa right. Here, yung OH nasa left, and then letter L, and then letter D. Ay, sorry kids. Namali og press. Now, as to the optical isomerism, yung letter D and letter L nyo, it is actually already related to optical isomerism, wherein your letter L stands for your levorotatory, and your letter D stands for your dextrorotatory. Now, an optically active compound is a compound that rotates the plane of a polarized light. And the presence of your asymmetrical carbon causes its optical activity. Now, what happens here in optical isomerism, that when your sample or a solution with with a carbohydrate present, when your solution may beam of plane polarized light, there is a beam of plane polarized light that is being passed through that solution with carbohydrates, a rotation will happen depending on what type of carbohydrate that is present. Yung definition natin kay D and L, na yung position ba ng OH, nasa left or nasa right, if dextrorotatory siya kids, yung ating carbohydrate, yung rotation niya, it will rotate in a clockwise direction, while for levorotatory, counterclockwise ang rotation or direction. So, yun lang yung tatandaan nyo. Again, for your dextrorotatory, clockwise direction. For your levorotatory, counterclockwise ang direction. And the reason why there is this certain optical activity, this rotation, is because of the presence of your asymmetrical carbon or your chiral carbon. So, yun lang tatandaan natin, kids. Huwag na natin pahirapan yung ating self. Next, we have your epimerism. So, here, it is the stereoisomerism if two monosaccharides differ from each other in their configuration around a single specific carbon only other than your anomeric atom. So, this is an example of epimerism. So, this is how we are going to state the difference or yung single specific carbon difference nila. So, base natin kay D-glucose. Now, if D-glucose is our basis, we could say that D-galactose, comparing it to D-glucose, is epimer at C4. Why? Because looking at the fourth carbon portion of your glucose and your galactose, diba, tingnan nyo, lahat-lahat same sila except dito na portion sa fourth carbon, wherein the position of OH of your glucose is on the right side, but for your galactose, it is on the left. So, yan lang po talaga yung epimerism natin. We are just looking at a single specific area wherein dun sila nag-differ. Or, comparing D-glucose and D-mannose, they are epimer at second carbon at C2. Kasi again, looking at the position of OH, kay D-mannose nasa left side, kay D-glucose nasa right side. So, ayun lang yung kay epimerism. Again, ang difference is yung single specific carbon lang. And the next for anomerism, These are isomers obtained from the change of position of your hydroxyl group attached to your anomeric carbon. Now, dalawa yung ating anomerism. It could either be identified as beta or alpha. So here, we are still looking at the position of your OH, but this time around, hindi po penultimate, but anomeric carbon. Now, so that it won't be that difficult for you to remember, kasi when we try to identify anomeric carbon, ito po kasi yung carbon na by the time we will transform your straight chain to a cyclic na form, yan yung portion kung saan makikita yung functional group nyo. But for you not to get confused, all you have to do is just to really check your first carbon. Kasi yan talaga yung basis din sa pag-draw sa cyclic structure ng mga carbohydrates nyo na kung saan yung functional group, that is where your first carbon will already be. So again, just look for your first carbon. Your first carbon will always be placed in this specific portion after kung saan yung oxygen nyo. And then look for the position of your OH. Now, as stated here, beta, kung OH nyo is on the same side of the ring. Or, it would be best to remember na kung beta siya, ito beta, meaning yung OH nasa taas. While for alpha, as stated here, lies on the opposite side of the ring, but for you to better remember, siya yung nasa baba. So, ganun lang yung tatandaan nyo. Now, for anomerism kids, let me emphasize that the only way for you to identify if it is beta or alpha is you should look at its cyclic na structure. Hindi po siya dapat dun sa straight chain. Cyclic structure po yung tinitingnan natin para masabi po natin na beta or alpha siya. So again, beta yung OH nasa taas, alpha yung OH nasa baba. So these are just some other examples. So as you can see, ito kasi kids, kasi yung functional group nito is ketone, and usually when you draw it in its cyclic form, naka 5 rings lang. But then again, dito pa rin po yung basis ng ating first carbon. So ayun, alpha nasa baba, beta yung OH nasa taas. And then here, this is an aldehyde group in which upon mag-draw kayo ng cyclic structure, then yung kinalabasan is a 6-sided ring. Then again, yung first carbon is andito pa rin po na portion. So again, alpha, baba, beta, taas. So next, we also have this type of carbohydrates. We call them your pyranose and your furanose. So still looking at your Haworth projection or your ring structure, pyranose, pyran would mean that it is a 6-membered ring, furan, 5-membered ring. Meron po tayong term na hemiacetal and hemiketal, and this is just referring to the functional group that is present. So na-explain ko na po kanina na yung 6-sided nyo, aldehyde ang present, and it is termed as hemiacetal ring. Yung 5-sided na ring nyo, it is furan, it is hemiketal, ketal kasi ketone na functional group ang present. So yan yung main difference ng cyclic structures ng carbohydrates nyo depending on the type of functional group that is present. And then we also have amino sugars. Diba, may mga monosaccharide din po tayo in which an OH group is replaced by your amino group. And very obvious kung amino sugar sila kasi the way we name our monosaccharides here will always end with the word amine. So ayan po, naka-highlight yung amine group nyo kung saan sila naka-attach in replacing what's supposed to be our OH, mga OH na group. So from your glucosamine, galactosamine, and your manosamine. Now, other reactions of monosaccharides that would end up having a different name, or we name this monosaccharides differently. Now, pwede na ay oxidation na mahitabo sa inyuhang monosaccharides. And when oxidation happens, it will produce acidic sugars. Now, it would greatly depend on what type of oxidizing agent it would be, if it is weak or if it is a strong oxidizing agent. Weak oxidizing agent, an example of that would be your Benedict solution. I'm pretty sure nakagamit mo Benedict solution in your laboratory experiments. For your strong oxidizing agent, best example for that would be your nitric acid. Now, if we used a weak oxidizing agent, then it will always end with onic acid. Ayan o, onic. If we used a strong oxidizing agent, then it will end with aric acid, aric. Now, with weak oxidizing agent, if yung oxidation, it happens at the aldehyde end of an aldose, then we call it aldonic acid. Or kung dun sa glucose tayo nag-oxidize, then we call it gluconic acid. Kung sa galactose, then that is what we call galactonic acid. As to your strong oxidizing agent, which can oxidize both ends of your monosaccharides at the same time, so it will produce a dicarboxylic acid or what we call an aldaric acid. For example, sa glucose nyo, it will be oxidized by your nitric acids, and then we call that your glucaric acid. So, sa naming lang din talaga yung point of emphasis po dito. Now, enzymes could actually also cause oxidation to your carbohydrates, kids, and it will produce uronic acid. So, that is why kung mapansin ninyo, ayan yung ending ng carbohydrate names nyo. You have your alduronic acid, or if specific na si glucose siya, then we call it glucuronic acid. So again, enzyme catalyzed oxidation yung uronic acids nyo. So here, it summarizes your monosaccharide derivatives because of the reactions that happened. So ito po for yung discussion natin kanina in terms of oxidation. So yung nakikita nyo na naka-highlight po, these are specific areas where the oxidation happened. And if you could still recall yung ating aldaric, di ba? This is what happened when a strong oxidizing agent oxidizes your carbohydrates. And as stated a while ago, diba, it can oxidize two areas agad-agad sa structure nyo. So, kaya may dalawang carboxylic acid na makikita dito na naka-highlight sa structure nyo. So, for aldonic acid, yung acidic group replacement is only at the topmost portion. For your alduronic acid, yung enzyme catalyzed, dun lang sa bottom part. For aldaric, both at the top and at the bottom. Now, reduction could also happen, kids. And when there is reduction of carbohydrates, what we will achieve are sugar alcohols. And that, yung ating naming is alditol. And alcohol groups on both top and bottom pa rin yung reduction na mangyayari. So, reduction to produce your sugar alcohols. Your carbonyl group is reduced to a hydroxyl group using hydrogen as your reducing agent. Drop the word O's and then change it to itol. So, kaya nga diba kanina na example, so ayun, if specifically si glucose yung ating i-reduced, then we will get the name glucitol. Glucitol is actually also known as your sorbitol. And sorbitol is a common sweetening agent used for your chewing gums. So here in this portion, kids, this is just to emphasize how important it is also to identify the presence of your monosaccharides or your carbohydrates, especially in identifying your different blood types. Diba, remember, we have 4 different blood types. You have your type O, your type A, your type B, and including your AB. Now, here, as of this moment, dili pa na akong upa-memorize on po ani kids. This is just to emphasize how important uh, identifying carbohydrates are, because it is one of the bases to identify what blood group system we are. But we'll leave this one na tipong dito na mo mag-memorize ug taman sa kung unsa man ni sila kids sa inyong blood banking na lang. So just to point out that itong mga nakikita nyo dito na color-coded, ito po yung ibig sabihin, and that your simplest monosaccharide lang po dito out of the 4 is your galactose. The rest are already monosaccharide derivatives. Diba? From your fucose to your N-acetylglucosamine to your N-acetylgalactosamine. Diba? From the word amine, so these are amine sugars. And then fucose is a special na parang carbohydrate. So, magdepende as to ilang galactose ba, ilang fucose ba, yung nakakabit sa red blood cell nyo. Basta again, that is the very basis for you to be identified as type O ba, type A ba, or type B ba. So, we are done with monosaccharides. Now, let us proceed with your oligosaccharides. Now, when we say oligosaccharides, kids, this time around, we are not looking at number of carbon atoms. We are now looking at monosaccharide units. Diba, as emphasized a while ago, lalo na dun sa portion sa hexoses nyo, na hexoses are the very need in terms of creating oligo and polysaccharides. So again, this time, we are not looking at number of carbon atoms, but the number of monosaccharide units. So when we say oligosaccharides, kids, that is around only 2 to 10 monosaccharide units. Beyond that, we will already consider it as a polysaccharide. And one thing that we need to know is your disaccharide. So when we say disaccharide, meaning dalawang monosaccharide unit joined together by what we call your glycosidic bond. So that is the specific name of the bond that exists between your carbohydrates. Diba? Peptide bond for your protein, glycosidic bond for your carbohydrates. Now, disaccharides are formed through condensation reactions between two monosaccharides, and upon hydrolysis, so of course, you will obtain your monosaccharides. So this is the name of your bond, your glycosidic bond. Of course, this time around, kids, dili na kuha nang magpa-identify sa inyo asa ang mga bond. So, humana na to. So, lahi na po na nga itong buhaton pohon. So, what are now your different types of disaccharides? We have your lactose, your sucrose, your maltose, and your isomaltose. So, let us start with lactose. So, as you all know, diba, lactose is very common and is present in milk, and it is the only sugar found in your milk. It is also synthesized by your mammary glands during lactation, and of course, it is used in infant formulations. And ito po yung combination ng lactose nyo. It is a combination of your glucose and your galactose. Now, what is present here in order to bind or combine your glucose and galactose is that there is a beta 1,4 glycosidic linkage. Now, beta referring to the position of OH. Now, again, it is found on top. As to your 1,4, kids, this is referring to the carbon atom asa specifically gibutang ang inyuhang glycosidic bond. So, 1 referring to the first carbon atom sa other ninyo na monosaccharide bound to the 4th carbon sa inyuhang other monosaccharide. So, kaya may 1,4. So, yan yung tinatawag na 1,4 glycosidic linkage. Next, how about your sucrose? Sucrose is the combination of your glucose and your fructose. And what exists here is a alpha 1,2 glycosidic linkage. So, one first carbon ni glucose and then combined sa second carbon ni fructose. Now, sucrose is commonly known as your table sugar, and it is one of the most prevalent na disaccharide, especially if we talk about dun sa plants. Now, it is obtained principally from the juice of your sugar cane, kaya sometimes it is called as your cane sugar, and it is also found in your sugar beets. Now, sucrose is one of the most common sweetening agent, lalo na kasi dun nga siya nakukuha sa sugar cane nyo. And then for maltose, this is combination of two glucose. And as you all know, pag maltose from the word malt, so meaning this is your malt sugar. And pag malt kids, 'di ba, pinaka ginagamit ang malt sugar ninyo sa pagbrew og beer. So, what is the difference between maltose and isomaltose? Now, actually, maltose and isomaltose are produces end products of your starch digestion. Differ lang yun ni sila kids in terms of the specific glycosidic linkage that is present. Why? Asa day ang kay maltose, asa day ang kay isomaltose. So as you've seen a while ago, for your isomaltose that is alpha 1,6, for your maltose that is alpha 1,4. So actually ang reason behind ngano na ay difference, ngano na ay maltose and isomaltose, it could be in the presence of, it could be because of the enzyme that is present. The way they cleave off your starch is in this way. Namo na natay isomaltose na makuha and naputay maltose na makuha. But then again, they are just both the same, especially in terms of their source, as far as I could recall, in terms of source and in terms of function. So they only differ in the glycosidic linkage that is present. Now, kani for sure na-experiment naman siguro po din ninyo siya as to knowing when one is a reducing sugar or not. Basta in terms of reducing sugars, consider all monosaccharides as reducing sugars and some of your disaccharides, especially your maltose and your lactose. So, kinsa ang non-reducing sugar sa inyong disaccharides, then that would be your sucrose. Now, for you to know if it is a reducing sugar, a reducing sugar will give a brick red precipitate on heating with your Benedict solution, and that the darker the color, the higher the amount of reducing sugar that is present. Now, remember that your Benedict solution is in this color, so therefore, when there is no color change, meaning to say there is no reducing sugar present, and as stated, the darker the color, the higher the amount of sugar present, then you might have observed these three different color. Diba? If medyo greenish siya, then traces lang. If medyo orange or yellow, then moderate. Or if nag-brick red na yun siya kids, then meaning taas kaayo ang reducing sugar na present ato sa inyuhang gigamit. Now, what is the reason behind why your maltose and lactose are considered as reducing sugar? Why is sucrose considered a non-reducing sugar? So, ito lang po talaga. It is really behind what could donate and accept electrons. Now, as stated here, maltose and lactose are reducing sugars because they have free carbonyl group which donates electrons that can reduce your katong copper na content sa inyuhang Benedict solution. Diba? So, ayan kasi yung free carbonyl group nyo. Diyan for your lactose, and then ito rin po for your maltose. While here, in your sucrose, wala siyang open end. No free carbonyl group is present to donate electrons kasi naka-attach kasi dun kasi yung glycosidic linkage nag-i-exist sa mismong specific portion kung saan yung functional group ni glucose and fructose. So, yun reason behind why we call them reducing sugars and non-reducing sugars. So, we are done with disaccharides. So, now let us go to your polysaccharides. So, what are polysaccharides? These are carbohydrates containing large number of monosaccharide units, each joined to the next by one or more glycosidic bond. So, since we defined oligosaccharide as 2 to 10, so here for polysaccharides, that is 11 up to pila na siya. Now, polysaccharides are also called as glycan. It is its alternate name. It is not sweet and it is negative in your Benedict's test. Siyempre, given that it is a very large unit, then of course, we would expect na lisod na jud kaayong mangita og free carbonyl na group sa inyuhang structure. Now, polysaccharides are non-sugars, insoluble, and high in molecular weight, and they function as simple source of energy, and they are also common as in the use of our making of structural materials, or basically dito pud na sila makita in terms of having ang function is more on sa structural na portion. And there are actually two types of polysaccharides. You have your homopolysaccharides and you have your hetero. When we say homo, only a single type of monosaccharide is present. When we say hetero, complex carbohydrates formed by combining carbohydrates with non-carbohydrate. And these are already examples of your homo and hetero. So under your homopolysaccharides, we have your starch, amylose, amylopectin, cellulose, and glycogen. For hetero, we have your pectin, chitin, and your heparin. So, unahon sa nato inyuhang homopolysaccharide. So, again, examples of homopolysaccharides are your starch, glycogen, and your cellulose. So, as mentioned, they are homopolysaccharides, meaning one common feature that these three examples have is that they are all composed of glucose. So, from our definition of homopolysaccharide, diba, only one single type of carbohydrate. And here, for your starch, glycogen, and cellulose, puro lang yun na sila glucose. Maski na moingon ta na naa sila over 100 ka monosaccharide units, those 100 monosaccharide units are all glucose bound together by your glycosidic bond. Now, one thing that could differentiate starch glycogen to your cellulose is in terms of the type of glucose. Kasi for your cellulose, it is in your beta, while your starch and glycogen, it is in your alpha. So now na lang sa put nato inyong cellulose having your beta-glucose residue. As you can see, about 10,000 chains of glucose, nakadikit-dikit through presence of your glycosidic linkage. It is the most abundant naturally occurring polysaccharide. It is the structural component of your cell walls in plants. It is fibrous, tasteless, odorless, and insoluble in water and most organic solvents. It is a linear polysaccharide of the glucose unit joined by your beta-1,4 glycosidic bond. Now, cellulose kids, one thing that is that you need to know about cellulose is that it is not an important source of nutrition for human beings because we lack the necessary enzyme capable of catalyzing the hydrolysis of this substance. So kaya As what we have discussed about cellulose, so naka-focus lang yun na sa iyaha sa plants. Next, as to starch, it is a polymer of alpha glucose and it is stored as starch grains it could be in your chloroplast, in your storage organs, or even in your seeds. Now, starch is used for energy storage in plants, and upon complete hydrolysis, it only yields the same as your cellulose na D-glucose din po. And that there are actually two types of starch. You have your amylose and you have your amylopectin. Now, what is the main difference between amylose and amylopectin? Looking at their structure, ito kay amylose, ito yung amylopectin, meaning amylose is linear, amylopectin is branching. Now, amylose being linear is less readily digested than your amylopectin, and it takes up less space than your amylopectin. Amylopectin is easy to break down by your enzymes to release glucose when needed because it is branching and it provides a larger surface area where your enzymes can act. Sometimes, the more branching ang inyuhang substance, the more na mas dali sila i-hydrolyze compared sa kung linear lang ang iyahang form. So, kana lang pod for your amylose and amylopectin. How about your glycogen? So, we've made mention that glycogen is your storage form of energy. Same with your amylose and amylopectin, alpha. Similar to amylopectin kasi branching din po yung glycogen but mas highly branch ang inyong glycogen. Ang branching occurs more frequently. So, compare it with your amylopectin here. Ito po si glycogen. So, ayan yung pagka-branch ng amylopectin nyo. And then, ganito yung branching ng glycogen nyo. Now, glycogen is stored by animals, chiefly in the liver and muscles. Not just animals, but us, human beings included. We store glycogen in the liver and in the muscles. And it is also stored by your fungi. So next, we go to your modified polysaccharides or katong inyuhang heteropolysaccharides. So this time around, in terms of their structure, it is not purely carbohydrates. It is not purely a single type of carbohydrate, but it is now in combination with non-carbohydrates pa. Like for example here, some carbohydrates are chemically modified by the addition of your functional group, just like your phosphates and amino groups, as seen here in your pectin and heparin. So let us discuss them one by one, starting with heparin. So I know you are already familiar with the word heparin because this has been discussed in your PMLS or maybe in your other laboratories, and that heparin is used as an anticoagulant. Other than that, heparin is actually also used as a drug, lalo na dun sa mga patients na need ng blood thinner. So it is widely used as an injectable blood thinner. It is also usually stored within the secretory granules of your mast or mast cells and is released onto your vasculature at sites of tissue energy. And for heparin, yung kanyang structure, you can find a sulfur or sulfate in its structure with only 15 to 90 disaccharide residues per chain. So, ayan yung sulfate sa structure niya. And then next, for pectin, it is a naturally occurring substance found in berries, apples, and other fruit. And when heated together with sugar, it causes a thickening that is characteristic of our jams and jellies. Now, other than that, pectin is a major component of your middle lamella. Lamella, kids, it is actually a parang tissue na makikita dun sa bones nyo. And lamella is actually the responsible in giving our compact bone its hardness and rigidity. So, yan yung role ng lamella. So pectin is a major component of your lamella, middle lamella, where it helps to bind cells together, but it is also found in your primary cell walls. And then next we have your chitin. Chitin is our second most abundant heteropolysaccharide, which gives rigidity to exoskeletons of your crabs, lobsters, shrimp, insects, and other arthropods. They are also found in the cell walls of your fungi, and it is used commercially in coatings to extend the shelf life of our fruits and meats. Now, the monosaccharide that is present in our chitin is your N-acetyl D-glucosamine. Looking at this structure. So, kaya siya tinatawag na N-acetyl glucosamine, kids, kasi itong ring structure, we call it your glucosamine. Itong white extended portion here, we call it your N-acetyl. And then last, we have your hyaluronic acid. Now, hyaluronic acid contains alternating residues of your N-acetyl glucosamine, included your glucuronate. It is a major component of our skin, where it is involved in tissue repair, and it is a major component of our synovial fluid, and was found to increase the viscosity of this fluid. So, yung synovial fluid nyo, yan po yung fluid that is found in our joints. So, ayan po yung itsura. It's just like your chitin. Chitin is purely N-acetyl glucosamine with hyaluronic acid at taaning glucuronic acid ninyo. And then, ingani na dayon na, kanina image, that is how your hyaluronic structure would look like. So, alternating your N-acetyl glucosamine and your glucuronic acid. So that ends our discussion for our carbohydrates, and our next topic for this Tuesday will be all about your lipids. So thank you so much for listening, and as mentioned last meeting, we will still pursue with your quiz since I was able to still give this video lecture ahead of time. Thank you so much, kids, for your patience. God bless.